Introduce myself, you already seen the name. So I'm Maitreyi Rajala. I'm basically virologist by training. Okay, my pre-doctoral, postdoctoral training, everything in virology. All right, today I'll be talking about influenza virus. So influenza virus is called the virologist's most wanted virus. You know, influenza virus, worse than SARS-CoV-2 virus. You know, and influenza virus in the past century, the world encountered several influenza viral pandemics much worse than SARS-CoV-2, okay, claiming millions of lives. So today I'll be talking about influenza virus. My main research focus is also on influenza virus only. So basically the virus and host protein interactions and how these interactions can be explored for the development of therapeutic drugs. Okay, the title says influenza A virus and host protein interaction, the potential drug targets. Okay, so I don't know, all of you are from biotechnology background? You're all biotechnology background? Okay, so you might have studied viruses somewhere in your bachelor's and master's, right? So for those who are not aware of viruses, much the structure, but with the SARS-CoV-2 virus, now the layman also understand the structure. They know how the virus look like. Well, so virus is a very tiny microorganism, submicroscopic organism. You cannot visualize virus under microscope, but you can visualize the effects caused by the virus under the microscope, right? Now this virus is the simplest organism. It's a submicroscopic organism, just composed of only two components. One is the genetic material, the other one is the protein capsid, right? But some viruses, they have an additional one, that is lipid envelope. Lipid bilayer, you see this one, it works like this only, the pointer. Pointer, it's okay. So here is this is the genetic material, and that orange one is the capsid. The purple one, what you are seeing is actually lipid envelope. That envelope actually the virus acquires from the host. It is not, because the virus doesn't have membrane biosynthetic machinery. So the envelope comes from the host, but that the pink and green ones you are seeing, the external surface proteins. These external surface proteins are viral encoded, not host derived, right? So basically how the virus replicates is not through division or binary fission or anything, but the components, if one virus particle enters the host, it can make hundreds of virus particles. So these components will be synthesized at different cellular addresses and they assemble into a complete virus particle. So we know virus is an obligate intracellular parasite, because the debate comes whether virus is a living entity or a dead material, okay? But if you say any living organism, it will pass on its genetic blueprint to another generation. So virus also does that. So taking that into consideration, virus is also a living entity. Now, why is it called obligate intracellular parasite? Because virus needs metabolically active cell. Only when the cell is metabolically active, the virus will be able to replicate and establish the infection, right? So because it doesn't have metabolic machinery, but it has the ability to manipulate the metabolic machinery of the host, but it doesn't encode any of those pathways, and it cannot produce energy, and it cannot synthesize its membrane or anything. So that's an obligate intracellular parasite. So some viruses, it has to depend on the host for replication, complete. But some viruses, bigger viruses, they can encode their own polymerase, okay, DNA polymerase. They encode their own RNA polymerase. So without host assistance, without utilizing the host DNA polymerase, still the virus will be able to replicate. But small viruses, they do not encode any enzymes, like replication enzymes are not encoded by these small viruses. Okay, they have to depend. But no matter whether the virus is small, encoding its own DNA polymerase and RNA polymerase, or a small virus, it doesn't encode any of those things, still the virus has to depend on the host for one thing, that is the translation machinery. Okay, virus doesn't encode translation machinery. Of course, the transcription machinery also, but the transcription machinery means the virus will be able to synthesize its own mRNA. Okay, but it cannot synthesize its protein. So for protein synthesis, translation machinery, the virus has to depend on the host, whether it is a small virus encoding only 3-4 genes, or a big virus like smallpox virus encoding more than 100-150 genes. Yet all the viruses, they have to depend on the host for translation machinery. Now, so these are the things, certain things virus encodes, and for certain things virus depends on the host. Like, replication of the genome, the virus has to have the information for its own replication, and the assembly and packaging, because when virus enters the host, there are host components also will be present. Host DNA will be present, host RNA will be there, host proteins also present. So how this virus makes its own particle, its own viral proteins. With its own viral protein, it has to construct the shell, protein shell. So that information should be present in the virus. They have the packaging signals. So the viral proteins will recognize the viral genome, not the host genome, and vice versa. And also, of course, the regulation, kinetics, and replication, etc. And it has to modulate the host defense. The virus is always will, devise certain strategies in order to escape the host immune system, so that it will be able to establish infection. But what are the things not encoded by virus yet the virus needs? One is the, as I already mentioned, translation machinery. Okay, translation machinery, membrane biosynthesis, it requires the membrane, and also it doesn't have any telomeres and centromeres. Now, this is just an introduction for those who don't know much about viruses. I'm trying to give. Virus is the tiniest organism. It's a submicroscopic organism. Some viruses encode the only 3 or 4 proteins, right? Then how if you see, every virus has to come inside, replicate, assemble, and into a complete virus particles will be released. So how does it do? Because it has to maximize its coding capacity. Some viral genome is only 3.1 kilobases, okay? Hardly anything. So, but that virus also replicates, undergoing multiple rounds of replication. Some viral genome may be much bigger, 100 kilobases or so, okay? But how do they manage? So there are certain strategies the virus will try to maximize its coding capacity. One is through alternative splicing, okay? Or some viruses, the viral transcripts will have alternative stop codons, okay? If it recognizes the first stop codon, it makes this much protein, and it ignores the first stop codon, moves on to the second stop codon, it makes this much protein. Likewise, it has alternative start codons, okay? When it recognizes the first start codon, full length protein will be generated, or when it ignores the first stop codon, moves on to the second start codon, it makes the truncated protein. So like that it does, and on top of that, it has to reduce the viral host gene expression, because the virus has to get the supremacy over host, because it hijacks the host machinery and makes it. So a little bit about the this one. So now the viruses are classified into 7 classes based on how they make the mRNA. So I think all of you know about David Baltimore, who discovered the reverse transcriptase enzyme. Recently only he died in September 2025, David Baltimore, Nobel laureate, and he actually identified the tumor viruses. It has a proviral DNA and it gets incorporated into the reverse transcriptase enzyme. It makes the reverse transcription. Okay, there are 7 classes. I hope you all will be able to read: double-stranded DNA, single-stranded DNA, double-stranded RNA, positive sense RNA. So influenza virus is RNA virus. Let me just explain about the RNA viruses. Okay, influenza virus is an RNA virus. RNA viruses, there are of 2 types. One is the one virus is a positive sense RNA virus. The positive sense RNA virus is basically, it is identical to the mRNA. Okay, so that means if the positive sense RNA is entering into the host cell, means immediately it will be translated into protein. If it is a negative sense RNA virus, negative sense RNA first enters into the host and it will be converted into positive sense, right? And then the positive sense, which is nothing but mRNA. I hope you are following. Okay, one is a negative sense RNA, the other one is the positive sense RNA. Positive sense RNA is nothing but mRNA. As soon as it enters the host, it will be translated into proteins. On the contrary, if it is an negative sense RNA virus, that negative sense RNA will be first it will be transcribed into mRNA, which is a positive sense. So it's written, mentioned, or it's not written. So positive sense, and that will be translated into proteins. So our interest, influenza virus, is a negative sense RNA virus. Okay. So one thing you need to understand, RNA viruses and DNA viruses. DNA viruses replicate inside the nucleus because DNA replication machinery is in the nucleus of the host. But RNA viruses, they don't have to go to the nucleus. Remember this point. They don't have to go to the nucleus. They can replicate inside the cytoplasm. Why is that? Can anyone say why RNA viruses will not enter the nucleus? The simple thing is, virus, it has to make RNA directed RNA synthesis. Okay? In other DNA viruses, DNA directed DNA synthesis. So for replication, DNA is the template. For RNA is the template for RNA viruses. And the RNA viruses, RNA dependent RNA synthesis. So for that, RNA polymerase is there. Is there in cells, do we find this one? RNA directed RNA polymerase? No. Right? That is a DNA directed RNA polymerase, right? So that's the reason. But this particular virus, now coming to the influenza virus. Influenza virus is a negative sense RNA virus, and this virus, though it is a negative sense virus, as I said, these viruses, they actually replicate inside the nucleus. Okay? Influenza virus, though it is an RNA virus, it replicates inside the nucleus, kind of against the law. Okay? So I'll tell you why is that when I talk about the replication. Now this is the virologist's most wanted virus is influenza virus. We encountered several influenza virus pandemics. No? Virus is not very far, it is always with us. Not like SARS-CoV-2, suddenly it came and caused a pandemic, claiming the lives. But this influenza virus, this is actually, I just got from this particular Nature paper, this figure, and this is 1918 Spanish flu. How many of you heard of this word Spanish flu, 1918? Spanish flu. This virus caused in 1918 Spanish flu, and this killed near about 15 million people, more than the people killed in World War. So this death toll and economic damage associated with flu highlight its role as one of the most harmful viruses ever known. Okay, so far we heard of many viruses in your lifetime. You must be in 20s. So you heard of Nipah virus, Zika virus, SARS-CoV-2, Ebola virus, and also any other virus. I think avian flu. You might have heard of avian flu and swine flu. Avian flu, swine flu are actually influenza viruses. All right. Now, this is the most dangerous virus, negative sense RNA virus, and it is all the time circulating in the population. Every winter, all of us get a flu, right? Mostly, either because of influenza virus, sometimes another virus, rhinovirus, is also a common causative agent of flu. All right. So, and not only that, this virus causes illness in humans, but also many animals. This virus is zoonotic in nature. All of you know what is zoonosis. Zoonotic in nature means this virus has the ability to cross the species barrier. So that means swine virus can infect humans. avian virus can infect humans. So this has the ability to cause the, to cross the species barrier. So this influenza virus not only infects humans but also infects several animals like cows, equine birds, aquatic birds largely, and also swine. Swine is the most culprit. I'll tell you why is that. And yeah, as I already said, it claimed 50 million, it caused 50 million deaths. And then after that, the seasonal flus actually, 2,90,000 to 6.5 lakhs deaths globally across every year, across the globe every year kills 2.9 to 6.5 deaths, right? So this is about influenza virus. And okay, so as I already mentioned, it infects most of the animals, cows, horses, dogs, frogs, salamanders, etc., etc. So mostly aquatic birds, avian strain, most of the dangerous viruses I'm going to talk about, the strains, they're all derived from the avian host, okay? From avian host crossing the barrier and infecting the humans. Generally viruses, there will be animal viruses, animal strains. For example, if you take papilloma virus, everybody is aware of papilloma virus. Papilloma has got bovine papilloma and human papilloma. Generally, bovine papilloma does not infect humans, the other way. And avian strains supposed to infect birds, and swine strains supposed to infect swine only, should not come to the human. Why is that? Why the birds, animal strains cannot infect the host humans? Because for the particular receptor is important, okay? That receptor is very important, a receptor that is present on the human epithelial cells. We're talking about influenza virus, which is a respiratory pathogen. So human lung epithelial cells, the receptor is different. The receptor being used by the human viruses is different from the receptor being used by the animal viruses. Even if we come in contact with the animal viruses, we will not be able to get infected because the receptor is not present. That specific receptor is important. But why from avian, from birds, how it is coming to humans? How from swine it is coming to humans, although there are different receptors. So we'll see that also. And of many potential subtypes of influenza, a few have resulted in serious outbreaks, I've already mentioned. If you see the H1N1, H3N2 strain, that's how the viral strains are designated, H3N1, H2N, N5, like that. What is basically H and N? H is the hemagglutinin protein which is present on the surface of the virus, and N is neuraminidase, okay? Neuraminidase protein which is also present on the membrane, okay? These are called surface protein, hemagglutinin and neuraminidase. Now the hemagglutinin protein is required for the entry. This protein has the receptor binding site. So through this hemagglutinin, the virus recognizes the receptor. And what is the other surface protein? That is the neuraminidase. Neuraminidase is the one which is required for the egress, okay? One is needed for entry. Hemagglutinin protein is needed for entry of the virus into the host, and neuraminidase protein is required for the exit of the virus from the host. Right? So there are so far, I think, I exactly don't remember, I think around 15 HA subtypes and 9 NA subtypes. They will be having combination.
13 and
9, okay. 13 HA subtypes and 9 NA subtypes, they would be exchanging. Okay, now these are the different pandemics we encountered. One is the H1N1, that is the Spanish flu, killing, it's written 20-40 million deaths, but elsewhere it mentions as 50 million deaths. And the next one is the H2N2, which is a new virus strain claiming 1 million lives. That is Asian flu and Hong Kong flu, avian flu, swine flu, avian flu, like that. So So these seasonal flus are common. They keep on moving, circulating in the population. But the pandemics are unpredictable. No, it is impossible to predict when we are going to have, but it is inevitable. We will definitely have the influenza pandemic. It can be mild, it can be serious like SARS-CoV-2 also, but is inevitable. Any time we can expect that new pandemic strain emergent. Okay, now this is the structure of the influenza virus, so you need to little bit understand for my talk. Okay, this influenza virus is that those red spikes, if you see these red spikes are, hemagglutinin, I guess, hemagglutinin, and the other one is the neuraminidase, blue one is neuraminidase, right? And also remember this structure, this is the virus nucleoprotein. Nucleoprotein is actually encapsulates the viral genome. Those balls, gray balls, is the nucleoprotein. The viral genome is a negative sense RNA genome, and if you see, there are segments. It is a segmented genome. Okay, this virus, influenza virus, has got 8 segments. Negative sense RNA segments 8. Okay, each segment encodes for 1 protein or maybe 2 protein. Okay, so there are 2 segments, that is M segment that encodes for 2 proteins, M1 protein and M2 protein, and another segment that encodes for NS1 and NS2 protein. So rest of them they encode for single protein. Together they encode around 17 proteins. Earlier 11, now the people they have shown, and some other proteins also their internal reading frames. Now the interesting part is this RNA virus. As I said, RNA virus replicates inside the cytoplasm, not in the nucleus. But this virus, being RNA virus, it enters the nucleus. Why it enters the nucleus? Because it has to use the host splicing machinery. Through alternative splicing, this M segment encodes for 2 proteins, okay, M1 and M2, and NS non-structural protein encodes for 2 proteins, NS1 and NS2. All right. So now each segment will be like this. What you are seeing here is this is how the segment. There you are seeing 8 segments. So each segment will be like this. With the gray balls are actually the nucleoprotein, and these are the PB1, PA, PB2. These are the subunits of the RNA-dependent RNA polymerase. All right. Now, another one here, you see that this white balls, and this is the matrix protein. So now I'll be talking on M1 protein and nucleoprotein. Now, the nucleoprotein is a capsid protein. M1 protein is actually, it is abundant virion, and this M1 protein gives rigidity to the virus particle. Virion. Okay, both are structural protein, but they're multifunctional. Along with the structural function, they also contribute to the replication. Especially nucleoprotein is needed for the replication of the viral genome, while the M1 proteins, which is also called as a matrix protein, and matrix protein is required for assembly packaging, right? And the neuraminidase, that neuraminidase, red color one is the neuraminidase, and the neuraminidase, as I already mentioned, it requires for the exit. And the hemagglutinin, that blue one is the hemagglutinin. Hemagglutinin is needed for the entry into the cell. Okay, now this is quickly, I'll tell you the replication. It is, I think it's a common one. It is like a general virus replication only. The only interesting fact is, though it is a RNA virus, it enters the nucleus because for 2 reasons. One, it has to use the splicing machinery. The other reason is, so the host mRNA will be there. Host mRNA undergoes the post-transcriptional modification. It has the cap and poly-A tail, right? Now this virus needs that cap, 12 to 13 nucleotide caps. The virus encodes for an enzyme like PB1, PB2, those balls. One has got the endonuclease activity. This virus, it goes, this particular protein, it cleaves the host mRNA cap, the 12 to 13 nucleotide, using that cap as a primer for the replication. Okay, using the cap as a host mRNA cap as a primer for the replication of the viral genome. So one is called cap snatching or cap stealing, and the other reason is a splicing machinery it has to. For these two reasons, although this is an RNA virus, it replicates inside the nucleus. Okay, now it is normal as usual. The virus and finds the receptor. It uses the sialic acid residues. Okay, so it finds the receptor. Through receptor-mediated endocytosis, the virus is internalized into the host. From there, the virus capsid will be disassembled, and all the 8 segments, 8 RNA segments, genomic is divided into segments, I said, and these segments will be translocated to the nucleus, where in the nucleus, using the mRNA cap, host mRNA cap, the virus replicates and then makes the progeny particles. And once the progeny particles are made, and then they are released outside through neuraminidase. Okay, through hemagglutinin enters, and through neuraminidase is the one which facilitates the exit of the new virus particles. Now, coming to the why this virus, what's the reason for epidemics and pandemics? You know, every year, annual epidemics, seasonal flus, and unpredictable pandemics. We don't know at any point of time the virus can emerge as a novel strain. You know, so the host cannot be, the pre-existing immunity cannot protect against that new virus. So what is the reason how the epidemics and pandemics occur? So because of the mutations, viruses are very fast. They mutated. The first thing is the virus is a RNA virus. Okay, RNA virus is it uses the RNA-dependent RNA polymerase. Now the RNA-dependent RNA polymerase doesn't have the proofreading capacity. Okay, DNA polymerase has the proofreading ability, will take care of it. So why do you think when the double-stranded DNA is more stable, why the viruses have evolved like that to have the RNA genome? That's basically to escape the immune system. They keep on changing, constantly they change. Okay, the first one is the antigenic drift. It is not a very dangerous mutations. Antigenic drift means slowly the mutations accumulate in the external surface proteins. Okay, so and then it makes a novel strain. Okay, some novel strain, I won't say, the segments will remain the same. So here if you see, it's coming, this is the normal one, then the mutations, the red dots are actually mutations. No mutation can, they can be certain mutations in hemagglutinin and neuraminidase. That's why they keep on changing, because neuraminidase and hemagglutinin, these are the external surface proteins. Those are the proteins exposed to the neutralizing antibodies. So they try to escape by changing their coat. The internal things are okay, but outside surface proteins will keep on changing in order to escape the immune response. So okay, so because of the antigenic drift, it is slow accumulation. But sometimes the segments will be exchanged suddenly. The segments will be exchanged, and then a new strain will be generated. Segments from, suppose a host. A single host cell is infected with 2 different types of viral strains, 2 influenza virus strains. As I said, it has a segmented genome. Because of the segmented genome, the segments can be swapped, can be exchanged. So when they exchange, that combination can be deadly. Sometimes it's just to go waste, but sometimes the combinations can be perfect combinations, and it can be very deadly and lethal combinations. And that is called the antigenic shift. The other one is random reassortment. If you see the color coded, okay, in random reassortment, this is the one drift. Because of the antigenic shift, sometimes they cause the pandemics, because the segments are exchanged. How the segments are exchanged? See, this is a bird, avian. Avian strain is supposed to infect the birds only, right? And that is the human strain. Human strain is supposed to infect the humans only, because for the human strain, human has got the receptor. For the avian strain, the bird cells have got the receptor. Now the swine should be swine, but actually where the farming takes place, poultry, The swine is the culprit because the swine has got receptor for avian strains, human strain. So that is called mixing vessel. Okay, so where the poultry and farming takes place, there, swine, if swine is infected with the avian strain and also with the human strain, and sometimes its own strain, there's a possibility. So there will be exchange of segments, and those exchange, if it is a perfect exchange, getting all 8 segments from some from one, then that will emerge as a very lethal, highly virulent and pathogenic strain, and that can cause pandemics. Now, say this is the one, the first one, this is how it goes. If you see here, now the Spanish flu from the aquatic birds, it came to humans. This is the most, all 8 segments, if you see, this one. This is the one, killed many people. Then after that, there's a combination took place from the another avian strain. If you see the red segments, okay, and the green segments, and they got here when they mixed up. So the new strain has got 3 segments from the avian, from the bird virus, and 5 segments from the human virus. So then after that, this is recombining with another avian strain, right, where you see the 5 from the human and 1 from the this previous bird already there, and other 2 are from another strain. Okay, and this is another one. Now the question mark. So there can be another combination also. That is why it is unpredictable. We don't know when this will, next pandemic influenza can occur. Okay, so any time it can emerge. Because of that is why it's very, because rest of the viruses are different. For example, SARS-CoV-2 virus, it is not segmented. It is a non-segmented viral genome, and that is a positive sense RNA virus, but only a positive sense RNA viruses, as soon as they enter the host, they will be translated into protein. If you take the positive sense RNA viruses, you just remove everything, remove the capsid. Protein shell can be removed. You just take that positive sense RNA genome. If you artificially introduce into any host cell, so the host cell will be able to make virus particles, because mRNA encodes for all the proteins that are required for the complete virus formation. So it can be, that is the reason polio virus is considered as a bioterrorism, not giving enough permission to handle the polio virus. Polio virus is a positive sense RNA virus. They have shown in a test tube making the polio virus particles. Okay, so anytime there can be another combination making, emerging as a novel strain. So this is what Dr. Zhang, manager of WHO's global influenza program, he says, another pandemic caused by a new influenza virus is a certainty. It is inevitable, definitely is going to cause. But we do not know when it will happen, what virus strain it will be, and how severe the disease will be. It is unpredictable. Anytime it can emerge. So what is the problem with the vaccines? Okay, there are certain influenza vaccines.
जब मतलब वायरस का आरएनए सेगमेंट के एक्सचेंज की वजह से जब न्यू सब टाइप हो रहा है, तो जो स्पेसिफिक टाइप ऑफ रिसेप्टर है ह्यूमन के अंदर, मतलब तो बाइंडिंग एफिशिएंसी तो कम होगा ना?
Binding efficiency कम होगा. Only it happens when the binding efficiency is strong. Okay, so that what happens? What question he asked, you understood? So suppose avian strain is mixing with the human strain. Okay. So human strain has got 1 segment which encodes for the hemagglutinin protein, right? That will remain. But rest of the segments will be exchanged. Not that, so the virus is changing, the host is not changing, right? Host is not changing, virus is changing. So if you, so suppose this is a hemagglutinin gene, okay, human. This will remain the same, but other segments it is receiving from the avian, right? So as a result, Hemagglutinin is normal. Hemagglutinin is still there, so it recognizes the receptor. Okay, so that combination, as I said, it should be perfect combination. If not, maybe there are some exchange takes place out of hundreds of virus particles, one may be successful in getting the lethal combination. So that can cause diseases. Now coming to the vaccines. Why the vaccines are so unsuccessful? Okay, now flu vaccines, but these flu vaccines are designed and developed every year. Because for example, we all take BCG vaccine only once, right? Now that is also stopped, but smallpox virus. Everybody heard of smallpox virus? Okay, I also didn't see in my lifetime, then what you will see, right? But the smallpox vaccine is so successful. Edward Jenner, he made that one. So successful. Why the smallpox vaccine is so successful? Now the smallpox virus is completely eradicated from the face of the earth. The reason is there is only one strain. Okay, only one strain. But if you take influenza virus, when it will change, nobody knows. Any point of time it will change. It will cross the species barrier. Okay, it will keep on changing. So the vaccines need to be designed and developed twice a year. Okay, so these vaccines, that is why the vaccines are actually, they're not very efficient, because the virus is constantly changing. So these pharmaceutical companies, they have to consult the WHO. They have to follow the WHO recommendations, and then WHO will recommend, okay, you make this strain, it can be divalent, tetravalent, something like 4 or 5 strains will be combined and the vaccine is made. So the WHO will recommend actually when to, WHO recommendations are used by national vaccine regulatory agencies and the pharmaceutical industries, and it will tell actually this year, this is the one, the virus is floating. In the population, you have to make the strain, this particular vaccine, right? So WHO last point, recommendations for influenza vaccine composition rest on the diligent year-round work for the global influenza surveillance. So global influenza surveillance will be there, and they will give the report, and these are the strains actually circulating this year. You better make the vaccine for this one using these particular strains, right? So that is why vaccines are not actually very efficient, and every year twice these vaccines are designed and developed, and the efficacy may vary from year to year, okay, lot to lot. So that's why vaccines are not very effective. So now see, this is the 20, 25, 26 season. These are the strains WHO recommended. Okay, these are the strains WHO recommended to make the vaccines. So likewise, every year, these pharmaceutical companies and national vaccine agencies, they have to listen to the WHO recommendation. They have to find out, then accordingly vaccines will be made. But these vaccines are not very successful. And what about anti-influenza viral drugs? There are therapeutic drugs are available, okay, and these drugs are basically against neuraminidase surface proteins. Okay, there are some against hemagglutinin also. I think there's one drug, I think hemagglutinin, but neuraminidase, external surface protein. That's what we discussed. External surface proteins are, they keep on changing. If you try to target, maybe it can be structure-based design, they will do structure-based crystal structure. If you know the crystal structure, actually neuraminidase inhibitor is made based on the crystal structure. So you are making the drugs, and that the catalytic site will be changed. Now the drug will no longer effective against that. So pre-existing immunity doesn't help, okay? So that's why it's a failure. Drugs actually, they're not very good. Flu antiviral drugs are prescription medicine that can be used to treat flu illness only. Actually, these drugs are given as a prophylactic. In 2009, I think all of you born after 2009 only. So 2009 as the swine flu. So swine flu pandemic. Neuraminidase inhibitor zanamivir, oseltamivir was given as a prophylactic drug. prophylactic means is just reduce the disease burden little bit, so the illness will be reduced, but only during early infection time. But later infection time, serious patients cannot be treated with zanamivir, okay? Zanamivir and oseltamivir. That's why it's given, this particular drug is given as a prophylactic, okay? So it is, drugs are not very effective. So these are the neuraminidase inhibitors generally given as a prophylactic drugs, like oseltamivir, paramivir, and zanamivir, and the endonuclease inhibitors are not that much given, and the other one is amantadine and rimantadine. Actually these were the first drugs identified by random screening. They just screen the chemical libraries, chemical compound libraries. That's how random. There's no rationale in designing that last drug, but neuraminidase with some rationale, because the structure is known, the neuraminidase structure is known, crystal structure. So using some rationale, these drugs were designed, but that is no rationale, just blindly random pick, and it worked. It worked against influenza virus ion channel protein, not against neuraminidase or hemagglutinin, against another protein, ion channel protein. Now actually these drugs are almost discontinued, not given, okay? Now the question comes, so with this vaccines failure, okay, and the drugs failure, okay, and there is one pandemic strain is waiting for us to meet us, okay? Anytime we may face that pandemic, right? So we have to prepare for the pandemic preparedness. Not that in the entire world, many virologists are working on influenza virus. So I'm also trying to do like, whatever I know a little bit, I'm trying to do that. But so considering this, ineffectiveness of vaccines and also the antiviral drugs, we need alternative approaches, okay? So all the time we are trying to target the external proteins, neuraminidase and hemagglutinin, but they are under constant change. They keep on changing, escaping the immune system. They will escape the immune surveillance of the host. Then we need some alternative therapeutics, alternative approaches, okay? What other approaches can be used? So while looking for designing certain drugs or using alternative therapeutic strategies, you have to take these points into consideration. What is that? Genomic variability. We have seen the mutations are common, they are accumulated in external surface protein, and because of the segmented genome, keep changing the segments, they swap, and then immune evasion. We know the virus can evade the immune system very easily because of the mutations, right? So taking these into consideration, we have to think about some innovative therapeutic approaches, okay? So when we design a drug, the drug should be having a broad efficacy, okay? Even we are infected by the avian strain combination, still that should be effective against, because different host origins from swine, from avian, equine, like that. So when we are designing a drug and developing a therapeutic strategies, and this should be able to target multiple strains, existing strains and also the emerging strain, okay? If we can think of something. So now the best one people are working on virus-host interaction, okay? So if you target the viral protein, most of the drugs are targeting viral protein. Now we know the viral proteins keep changing. If we target the host protein, okay, if you try to knock down the host protein, that host protein, it may be lethal to the host. That host protein may be having an important function in the host, okay? So if you just target the host protein, it can be a problem to the host. It is like instead of killing the virus, you kill the host, right? Something like that. So you cannot do that, okay? So the best way is to target the interactions, because the virus is an obligate intercellular parasite. The virus cannot replicate on its own. The virus has to, the fate of the virus depends on its interaction with the host proteins. The virus interacts with the several host proteins, and these host proteins will determine the fate of the virus. Of course, that interaction only will determine the fate of the host also, right? It is like a battle. There's a battle between the host and virus. So, the virus definitely it will mount some defensive strategies, but the host is smarter than virus. And this virus actually come up with a different strategies in order to evade the host immune response. So now the virus being an obligate parasite, it uses the host machinery. Okay. Now identify what are the interacting partners. Okay. If we can identify the interacting partner, not just identifying the interacting partner, but also identifying the regulatory mechanism of that interaction. If a viral protein is interacting with the host protein, so what is the function? Now virus doesn't do any extra burden, virus won't take anything. You know, if it is doing means there is some requirement, the virus has to regulate some mechanism in the infected cells, right? So new line of understanding of virus host interaction is the PPI network. Some of you must be having with the computational biology experience in bioinformatics, also using the computational biology tools, and then we can identify the protein-protein network, and you try to understand that interaction between the host protein and the virus protein. So identifying drug targets, then you identify the drug target. There you see influenza host protein interaction, viral pathogen, it is like a seesaw. It goes up like this. It depends on the host immune system. How good is the host immune system? How efficient is the virus? Now, host influenza virus host interactomes as a basis for antiviral targeting. So, in order to identify the antiviral targets, so we have to understand the viral and host protein interactions. Because the viruses mostly, for example, you cannot target DNA polymerase of the virus. Okay, if you try to target the DNA polymerase of the virus, and probably the host DNA polymerase also will be affected. Right? So it's a very important which viral protein you are choosing, which host protein you are recognizing. If you knock down or if you target that host protein, it should not have much of a biological implications on the host. For example, HIV. HIV, everybody knows HIV retroviruses. Now this HIV, there is one drug, Maitreyi drug. This Maitreyi drug is actually targets the host receptor. Generally, we cannot target the host proteins. We have to target the viral proteins. But Maitreyi drug actually targets the receptor, the host receptor. HIV uses a co-receptor CCR5. So this CCR5 is there is a drug which targets the CCR5 because they have shown if you disrupt whatever, if you target the CCR5 receptor, it doesn't have a biological implications on the host. So all that you have to take into consideration. Right? Taking all that into consideration, you have to identify the host factors, host dependent factors, okay, for the virus. So taking that, we have to design. So my lab works on influenza virus, two proteins, matrix protein and nucleoprotein. Nucleoprotein is a structural protein, capsid protein. You have seen that gray balls, it is encapsulated. There is a nucleoprotein that round inside those one layer, that is the matrix protein. So this is what I have taken the influenza virus. I have shown 10 proteins. Actually influenza encodes for more than 10 proteins, but these are the major proteins, and I have taken nucleoprotein, M protein, matrix protein. Okay, nucleoprotein is a structural protein, matrix protein is also structural protein, right? But they are multifunctional. They just regulate multiple functions in the host cell. Okay, not just structural function, but also they have other functions like replication, assembly, packaging, and this nucleoprotein shuttles between the cytoplasm and the nucleus. The shuttling is very important. Okay, so I have checked, and another reason for why I have taken this is both the proteins, they are highly conserved proteins. They're not changing protein like neuraminidase hemagglutinin. So they have 90% sequence similarity with the swine, with the avian strains also. So the possibility of changing is very less, right? So that is why I have chosen these two proteins. Right now we don't have any drugs against nucleoprotein and matrix protein, but I just want to work and why this nucleoprotein, what are the protein, what are the interacting partners of the viral nucleoprotein and also the matrix protein. So we have done mass spec analysis, the first experiment. So quickly a little bit about the nucleoprotein. And this nucleoprotein is a structural protein conserved among the strains of different influenza viruses, 90% sequence similarity, and there is a major protein of the ribonucleoprotein complex because positive sense RNA virus is like a naked. It is just like that it comes. But if you say take the negative sense RNA virus is always coated with some proteins. It forms the ribonucleoprotein complex. Why? Because it needs some time from negative sense to it has to be converted into positive sense. During that there is a possibility it will be chewed up by the RNases. It's like a protection. So actually this nucleoprotein is also kind of a protecting that. It encapsulates the viral genome, negative sense RNA genome, right? And then it forms the viral ribonucleoprotein complex. And also it shuttles between the cytoplasm and the nucleus, because I said although this is an RNA virus, it replicates inside the nucleus. So this nucleoprotein will be shuttling because it performs multiple functions. The replication of the viral genome, nucleoprotein is very important. Okay. And so we have done a mass, I'm not, just briefly I brought some slides to show you. Yeah, we have done mass spec analysis. The first thing I just wanted to see what are the host proteins that are likely to be interacting with nucleoprotein. Okay, so we have done 2D gel electrophoresis and mass spec analysis, and I have identified nucleolin. Nucleolin is actually largely reside in the nucleolus and also in the nucleus, and actually it moves from the nucleus, nucleolus, cytoplasm, and sometimes it goes to the membrane also. Then we identified by mass spec analysis, we identified nucleolin. Remember, nucleolin is the host protein, nucleoprotein is the viral protein, right? So then we identified nucleolin, a multifunctional eukaryotic protein, was identified to be one of the interacting partner. I have identified many, so one by one, few only I'm going to tell you here. So we identified nucleolin as this is also multifunctional protein, and this is also largely reside in the nucleus, right? And then this is the interacting partner, and this nucleolin it has a role in pre-RNA, rRNA transcription, transcriptional elongation, and ribosome assembly. And largely, as I said, it locates in the cell nucleolus. Okay, so the first experiment I have done, because by mass spec analysis, 2D gel and mass spec analysis, we identified those peptides, the nucleolin. So now we have to confirm whether the nucleolin is interacting with virus nucleoprotein or not. So I have used various protein-protein interaction techniques, do you know any of the protein-protein interaction? What? SPR? Is2 hybrid system. Any other techniques? Immunoprecipitation, co-immunoprecipitation, we can say. Size exclusion chromatography will tell us FRET, yeah, and microRNA won't tell us the protein-protein interactions. Okay, colocalization experiments by confocal microscopy, right? So we have checked this one by I think I don't have to explain. These are the left side are the immunoprecipitation assays. We pull down with one antibody and we probe it with the another antibody. And then if you see both the proteins, then you know the complex. In a pull down experiments, you will find the complex. That complex can be, it can be subjected to SDS page followed by Western blot analysis. You will be able to see. See here, you have seen the nucleolin. Once you pull down with the nucleolin antibody, you see the nucleoprotein antibody. In a reverse manner, you do it. Pull down with the viral nucleoprotein antibody, and you do look for the nucleolin. Likewise, I have identified this protein as actually interacting with the nucleolin is interacting with the nucleoprotein. Rather, viral nucleoprotein is interacting with the host nucleolin. Then we just wanted to check by the recombinant constructs. I made a recombinant construct of the nucleoprotein with green fluorescence tag and nucleolin with red fluorescence tag, cherry tag, something we had put. So we have seen here, you can see the green one is the recombinant nucleoprotein and red one is the nucleolin, recombinant nucleolin, and you can see the merge. Okay, that merge will tell you the co-localization by confocal microscopy, but the co-localization will not tell us that these two proteins are physically interacting. The complex, they are there in the cell, they are together. You know, that yellow color, it shows that they are co-localized. But this co-localization will not tell us they are physically and directly interacting. There may be some intermediate player also. Right? So anyway, here we have seen that this protein is, we can see the merge, and then these two proteins, this confirms that by co-immunoprecipitation assay and confocal microscopy, we confirm that the nucleoprotein interacts with the host nucleolin. Then that we did with the recombinant proteins. Okay, recombinant protein. Now we have to see with the real virus. Okay, so is the same thing happening in virus infected cells or not? First we just check it, we made the recombinant construct. Now we need to do the same thing is happening inside the cell or not. The cells infected with influenza A virus. I work with 2009 pandemic swine flu. Okay, and also I validate all the experiments with the regular floating strains, seasonal strains, they are called H1N1, H3N2 strain. Whatever I'm seeing in the pandemic, I should be able to see in the seasonal strains also. Then only we will be able to target. If it is restricted to only pandemic strain, if we try to develop any therapeutic approach or anything, probably that will not be able to target the seasonal strains. So we need to actually, when you are handling with the pandemic strains, you have to check the same thing is happening in seasonal strains also or not, cells infected with seasonal strains. So this I have checked with the virus infected cells. You know, lung epithelial cell line is infected with, because the virus is a respiratory pathogen, it infects the lung epithelial lining. So we have taken the lung epithelial cells and we have checked. Yes, you can see the co-localization there also. You see by co-immunoprecipitation, we have seen this nucleolin as a interacting partner to the novel interacting partner to influenza A virus nucleoprotein. And in the same way, one more experiment, one more confirmation we have done by the in vitro binding assay. In vitro binding assay, you know what is in vitro binding assay? Okay, we make the recombinant construct, one of the proteins. Now, right now we have two proteins under question. One is the viral nucleoprotein, the other one is the host nucleolin. So what we do is we just make the nucleoprotein construct. Okay, you express in BL21 cells, in a prokaryotic vector, you put it and express, then put your A549 cell, lung epithelial cell line. Okay, cell lysate, because the lung epithelial cells have the nucleolin protein, whether these two proteins are interacting or not. That is the simplest method we do beginning. As an in vitro binding assay we have done. So I want to check. So now it's confirmed by 3 or 4 different experiments that nucleoprotein is interacting with nucleolin. So what is its function? It has got any regulatory role. If I knock down nucleolin, what will be the fate of the virus? Okay, so when we knock down, you have to take care. So if you knock down nucleolin, host nucleolin, they should not have some impact on the host cell. There's a possibility it becomes lethal. the cell might die. So you have to take it, you have to consider that point also. So we have done siRNA method. I think all of you know siRNA technology. Using siRNA method, we have seen, we have knocked down the host nucleolin. See, nucleolin, this is the non-targeted siRNA. So nucleolin levels are normal, but it is knocked down, and by the Western blot analysis also it's confirmed. You don't see the siRNA nucleolin. So when we knock down nucleolin, we see the reduction of the nucleolin. Then what happens if I knock down, remove, reduce the nucleolin levels in the cell and infect and do it, because the virus is not finding enough nucleolin to interact. Is that right? When you are silencing it, then what will happen to what is the fate of the virus? Then I have seen actually, when I knock down this, silence the nucleolin, actually the virus replication goes down. Okay, the virus replication reduced to 100-fold tighter. So what is that? It is actually the host is playing a defensive role through nucleolin interaction. When nucleolin interacting with the nucleoprotein, actually the virus replication goes down. Okay, that is why if you knock down, you disrupt nucleolin. So that means you are disrupting the interaction between the nucleoprotein and nucleolin, right? What will happen to the virus? So you see the viral replication goes down, increases. Okay, so that means nucleolin interaction is not good for the virus. Okay, it is a defensive mechanism. So if you knock down nucleolin and it goes down, you got it? Then I thought, okay, then let me see, what if I overexpress nucleolin? Okay, so what should happen? If I disrupt, knock down a nucleolin, virus replication goes very high. If I overexpress nucleolin, what should happen? Host will be protected, and then see, it is gene expressions I have checked, it comes down. Do you see that one? Okay, so, and here also, and this is with the whatever I have done with the pandemic strain, I have checked, confirmed with the seasonal strains also. In seasonal strains also you see the merge, the nucleoprotein and nucleolin, both of them are interacting, not only in case of a pandemic strain, but also with the seasonal influenza virus nucleoprotein. So this tells, okay, nucleolin is not actually desired by the virus. The virus doesn't want nucleolin. If nucleolin is present, but early hours, but as I said, the virus is smart. But after some time, it will try to overcome the obstacle, because the host is always try to defend itself against the invading pathogen, right? But the virus is much smarter than the host, and it will try to come up with different it will devise several strategies to escape that. But anyway, we have seen the nucleolin is playing a defensive role. So upon interaction, when nucleolin interacts with the nucleoprotein, the virus replication is limited. Okay, if you overexpress nucleolin, actually replication goes down. So then, so it tells this, the interaction between the virus nucleoprotein and host nucleolin, it has some regulatory role. Okay, so then I wanted to do the comprehensive analysis where the virus to exactly what is this replication. I wanted to check at the domain wise. So let me take the domains, which domain of the nucleoprotein is actually interacting with the nucleolin, right? And what are the residues, amino acid residues that are critical for interaction between the host nucleolin and virus nucleoprotein. And also I don't know, because I have done the co-immunoprecipitation assay, confocal microscopy experiments I have shown earlier slide, but these two experiments will not tell us that they are physically interacting or not. Okay, they may be in the complex, that's why they are co-localized. There may be some intermediate player also, like this is, let's take A and B. So maybe some C is there A is interacting with C and B is also interacting with C, then still you will see the co-localization. So that's why we wanted to see to develop any therapeutic approaches. Okay, so we need to understand whether these two proteins are physically interacting or not, to block that interaction or to target that interaction. Then we have done individually gel purification, we have done all that, nucleoprotein and nucleolin, we have done everything, and then we have done the SPR. As he said, the surface plasmon resonance, it will tell these two proteins are physically interacting and directly interacting. Then I wanted to see what are the domains. Okay, the nucleoprotein has got 3 domains, RNA binding domain, so that you see this one, 1, 2, 3 domains. So I just wanted to see which domain is actually interacting with the, not the entire protein will interact, maybe certain residues. So I wanted to say which domain is actually interacting with the nucleolin. So we generated deletion mutants like GFP, GFPNP, each one, but we had put the tag. It's easy for us to visualize. Green fluorescence tag is getting expressed or not. So you putting the green fluorescence tag, we made the domain-wise deletion mutants, and then we checked one by one. When we checked, actually, so all these different expression levels we have seen, and we have checked actually the RNA binding domain and homo-oligomer. Do you see the second one? You may not be able to understand this one. This is co-localization, it tells. So if you see the right one, see the first domain, second domain, third domain, other one is the full length. See here, this is the first domain, RNA binding domain, is immunoprecipitation assay, and this is the second domain, homo-oligomerization domain, and this is the homo-oligomerization domain as a full length. Okay, so by this experiment it tells most likely the nucleoprotein interacting with host nucleolin through RNA binding domain and homo-oligomerization domain. Okay, so it tells. Then we wanted to check residues, critical residues. So what are those residues? If I can disrupt that residues or not. So we have, because these two proteins, the swine flu nucleoprotein, crystal structures are not available. Even nucleolin crystal structure, full length crystal structure is also not available. So this host nucleoprotein and the nucleolin and viral nucleoprotein, we just developed the 3-dimensional models. Using the 3-dimensional models and in silico docking was done, and when we identified certain residues. So it's given in this one. These are the, I'm sorry, those images, yeah. These are the residues actually. We have done at protein level, they directly interact, done at domain level, okay, and this is there, and this is actually, these are the residues, okay. So nucleolin, here it tells, yeah, so here we are seeing the co-localization, all that, it tells the interaction, that interaction is through critical amino acid residues. Okay, 5 residues were identified. So I want to understand are the 5, all the 5 are important or one is important. Now 1 we are trying to disrupt, 1 amino acid, 5 residues been developed. So 1 we are deleting, and then we made all the mutants, different mutants, nucleoprotein, those are deletion mutants, these are individual, only 1 residue we are disrupting, and we are actually working on this one. This work up to here, we just published one in the previous one, nucleolin interaction also we published, and this is also we published. The most interesting part is whatever amino acid residues we identified to be critical for interaction, and these residues are conserved across the past pandemics. No? Because the nucleoprotein is a conserved protein. So all these residues were identified to be conserved in the past pandemic strains. That's a one good encouraging results. Now we are working and in collaboration with anybody is from Central University of Punjab? Okay, collaborating with your university only, Central University of Punjab, we are trying to understand all the, okay. Now the next one is a matrix protein, right? The matrix protein and another abundant protein. I'll stop here, that nucleoprotein story. Let me quickly tell you about the matrix protein. Matrix protein is the inner ring which is beneath the lipid bilayer. Okay? This virus is an enveloped virus. So this rigid protein, this protein is under the lipid bilayer, and this protein gives rigidity to the virion. And interesting, this protein I have chosen because this protein is also 90 to 95% sequence similarity with the swine, avian and human, because usually combination takes place among these swine, avian, not equine, very less, but usually swine, avian and this one human. So we have taken 90 to 100% amino acid sequence similarity. So we have taken the matrix protein, and we identified what happens if a matrix protein, that recombinant matrix protein, when we express, what will happen to the cells, what are the cellular proteins that are differentially upregulated. So we done the mass spec only using the matrix protein and nucleoprotein also I checked, and then expressed, and I tried to identify the differentially expressed proteins. So interestingly I found with both the circled one, alpha enolase, pyruvate kinase, there also with matrix protein alpha enolase and pyruvate kinase. You must be saying the difference is not that much, why you have chosen alpha enolase and pyruvate kinase. There are some large difference like HSP70, a big difference. What is that? RAS related protein, big difference. But if you see alpha enolase and pyruvate kinase, they don't have much difference. The difference between the mock infected cells and because every time we have to put the control. Okay, uninfected cells, mock infected cells, and when we see in both, in response to matrix protein and in response to nucleoprotein also, we have seen the differential regulation of alpha enolase and pyruvate kinase. When we identified this thing, more or less at the same time, a few years before, somebody published a paper saying that this alpha enolase and pyruvate kinase. What are these? These are glycolytic enzymes, right? Now, alpha enolase and pyruvate kinase, they identified inside the virus. Okay? Usually, when virus is assembling and packaged into a complete virus particle, it takes only its own proteins, viral encoded protein. It doesn't pack the host proteins. Okay? It is like an extra burden for the virus. Virus will never take the extra burden generally. So, they identified in a purified influenza virions, actually they identified alpha enolase and pyruvate kinase. So earlier it was thought maybe by accident, while it is packing its own protein components, maybe by mistake some of the host proteins also being packaged. But later the people started understanding there is it's a meaningful thing. For the virus does that. So they have published in a paper like this virus packs the alpha enolase and pyruvate kinase. So I got interested. I said, what is this? Well, alpha enolase and pyruvate kinase, how they go? As I said, there is a packaging signals. The viral genome will recognize viral proteins only, not the host protein. Then how come the host proteins are going inside? They cannot jump and go into the virion. So logic thing is the external protein, either it should interacting with the nucleoprotein or matrix protein. So we have done that. So this also we could communicate this one. So we have checked, and I'm not going into details, all in vitro binding assays and everything we have done, and immunofluorescence, immunoprecipitations, etc. Actually, alpha enolase and pyruvate kinase, they nicely interact with matrix protein and nucleoprotein. Okay, so there is something. This is another interaction. So can we target that? So because we know the viruses, in order to meet their energy demands, they have to manipulate, reprogram the metabolic machinery. Viruses can manipulate the metabolic processes in order to meet the energy demands, because there is no energy synthesis or anything they cannot make, so they have to do it. So, and these are simple immunoprecipitation. I am not going to explain. We have used all the constructs in vitro, like prokaryotic expression vector, and also in eukaryotic expression vector we made all the constructs of nucleoprotein and matrix protein, and then we have done, yes, they interact with, see here, they interact with each other. Okay, and now this interaction is also direct or not, I don't know. Somebody from Central University of Punjab is coming to do, I think he's here to do summer training here. Probably I decided to give this topic to him. So because it tells like they interact, but it is a direct, or what are the other cellular proteins in the complex, we are not, it is not known. So all these experiments have done. Okay, and next thing, just I wanted to see what happens, what is the level of, I didn't bring those images, alpha enolase when virus infects the cell. When virus infects the cell, what will happen to the alpha enolase and pyruvate kinase? We have seen alpha enolase and pyruvate kinase, they interact with the nucleoprotein and the matrix protein, but the regulatory mechanism I wanted to say, because of that viral infection, is the alpha enolase and pyruvate kinase levels are fluctuating or not. When I checked, actually no difference. So the viral, this alpha enolase and pyruvate kinase expression remains the same, comparable with the uninfected cells. Then what is it? Then we check the activity. I wanted to see the alpha enolase and pyruvate kinase activity. Yes, in infected cells the alpha enolase and pyruvate kinase activity goes high, and when I inhibit the activity with some inhibitors, there are some inhibitors like chikungunya and Enoblock, PKM and alpha enolase inhibitors. So when we inhibit, actually the virus replication goes down. Do you see the picture? Above one is those blots, above one is pyruvate kinase, the below one is the alpha enolase. So when we treat in the presence of, these are mock infected, this is virus infected cells in the presence of drugs, alpha enolase activity inhibitor and pyruvate kinase activity inhibitor, then we see the viral gene expression goes down. Okay, that means this alpha enolase and pyruvate kinase is playing some role. This interaction is regulating the expression of the virus, but sometimes these viruses, these metabolic enzymes can play the non-metabolic role also. So now I cannot say whether these are playing a metabolic role or not, but definitely the alpha enolase and pyruvate kinase is doing something through their interaction with the viral proteins. And then we wanted to check the glucose metabolism, whether the glucose uptake is increased or not, because these are involved in glycolysis, alpha enolase and pyruvate kinase. So I wanted to see the glycolysis and glucose uptake and etc. So we have seen actually early hours, all these events take place during early infection, that is within 6 to, because the virus takes 6 to 8 hours to complete one round of replication. During initial stages only it will try to set the platform for its beneficial outcome, so that it can just take care of everything, so that the virus will be able to replicate more efficiently, will be able to establish infection in the host. So we have seen actually the glucose uptake is increased. Then when the glucose uptake is increased means, just we wanted to see the other picture, I think I haven't brought. Okay. Then we saw when glucose uptake is increased means how it is doing. So only in the presence of, when we express the nucleoprotein, recombinant nucleoprotein and recombinant M1 protein, the glucose uptake is going on. So glucose uptake is going on means probably glucose transporters needs to be increase the expression. We check that the glucose transporters, if the glucose transporter level is increasing, only when the glucose transporter level increases, and then we'll be able to take the glucose. Then we have seen, see very nicely, these are M1 protein. The red one is the M1 protein and the green one is the GLUT4. Actually the M1 protein is upregulating the GLUT4. It doesn't have any effect on the GLUT1 right? But M1 protein, we see very nicely, see this one, and not only that, they are colocalizing. Okay, so M1 protein is likely to be regulating the glucose transporters. When the glucose transporters 4 is increased, not glucose transporter 1. So when glucose transporter 4 is increased, actually the glucose uptake is increasing, and the glucose uptake is increased means there is a possibility the glucose metabolism, either glycolysis or may enter in another pathway also, that we are seeing into that. And not only that, if you see that this last one, on the contrary, there you are seeing increased glucose transporter 4 expression in virus infected cells. When you take independent of other viral proteins, only recombinant matrix protein, see the glucose transporter 4 level is decreased. Now these two experiments will tell, though I don't know what exactly is happening here. So see here, and the M1, when the glucose transporter expression is decreased, you see this one, and sorry, when it is decreased, the virus gene expression is also being decreased. Okay, so the glucose transporter 4 is important for uptake of the glucose. So when you inhibit this one, when it goes down, and the viral gene expression is also going down, this is it. And then we have seen like this. So we thought, is it through glucose transporter 4, the virus is modulating the uptake of glucose in infected cells? What should happen when we just block the glucose transporter 4? If you inhibit the glucose transporter 4, we should be able to see the reduction of the viral infection, right? When I am saying the M1 protein is regulating GLUT4, then if you reduce the GLUT4, the expression has to go down, the viral replication has to go down. So we have exactly seen, we have used the glucose transporter 4 inhibitor indinavir, when we used actually not only that glucose uptake reduced, we are trying to block the GLUT4. So glucose uptake also reduced. With the reduction of the glucose uptake, viral gene expression was also reduced. So with these experiments, a few images I hope you followed. So these findings, so we identified other one also retinol dehydrogenase, and another one we identified. So one is that the most important ones we have seen here is the interaction between the viral nucleoprotein with the nucleolin, and interaction between the matrix protein with the alpha amylase and pyruvate kinase and the glucose transporter 4. So these findings would tell, actually it is giving us some insights into the binding mechanisms. That means, and we have to study in detail also, okay? So to develop some alternative therapeutic approaches instead of targeting the viral protein, but most of the antiviral drugs target the viral proteins, but they are changing, and we cannot directly, I cannot target the alpha amylase and pyruvate kinase, very important. So we can target that interaction. So how you can target that interaction? For example, nucleolin. I am saying nucleolin is playing a defensive role. Okay, so by promoting interaction between the nucleoprotein and nucleolin, actually virus replication goes. So what we can do? So we can think of some peptides, they mimic the nucleolin. We can design certain peptides, they mimic the nucleolin, they will be able to interact, and the function of the nucleoprotein may be inhibited. So here these findings suggest the importance of interaction between the viral and host protein interaction, and provides an insight into the binding mechanism involved in the interaction. This is the most important ones. So it is like a tug of war. So if you see this image, virus would be winning, then the host think it will change, and accordingly virus will change. See, it's just illustration it's given, and so first one is like a sharp one, and it gets the round one, and the virus also gets like that. It is like, it will keep on changing. It is like it's a war between these two ones. So now the virus-host protein interactions is an alternative therapeutic approach to develop anti-influenza drugs. So because a comprehensive overview of viral protein participating in key stages of the viral cycle can help understand the molecular events that is, the virus is regulating. Okay, so basically you can target any step. You can target at the entry level, or you can target within the cell replication, or you can target at the exit level also. It is up to you. If you identified interaction, we don't know exactly where it is playing. So that's what we are trying to understand, exactly whether it is playing some role in packaging or replication or assembly. All that we need to do it. So, but you have to understand very clearly the comprehensive analysis, what are the residues, how they are interacting, all the information is needed in order to disrupt that protein-protein interactions. So it can be a better therapeutic approach. For example, because these viruses, if you see the glycolytic enzymes, glucose metabolism and all that, the viruses, they're known for it. Because they do not encode for any metabolic machinery, they just try to manipulate, or otherwise they reprogram for their own benefit. The viruses can reprogram the host metabolic pathways for its own benefit and for beneficial outcome also. So these things we can understand. So basically we cannot block the alpha amylase and pyruvate kinase, but transient. If you can transiently block the glucose just for a short period, probably that will not have too much impact on the host. So transiently those pathways can be blocked, and so that you can limit the virus replication. Okay, so like that the interaction, because every virus has got some host dependency factors. If you can identify those host factors which are required for the virus replication, if you can just disrupt that interaction, probably will be able to reduce the disease burden. Okay, here you can see that the purple one and red, that interaction can be. Okay, we can do lot of things, interactome, like PPI network, you can do it. Then after that interactions can be, as some of you said, like a protein-protein interaction can be studied by SPR and Easter 2 hybrid system. Several other techniques are there. Then after that, you look for what is that regulatory mechanism. When you identify two protein, one virus and one host protein, they're interacting with each other, what that interaction is regulating in the regulating. Okay, so all that can be mechanism of infection, identification of new target, and pathway and mechanism triggered by multiple pathogens, disease etiology, like that. All the things can be studied. So this is what, either entry level you can do it, or at the exit level. See, while it is coming, you block that one, or while going out, you block it, either way. So right now there are these are called host-directed antiviral strategies, because you are identifying the host factors interacting with the viral factors. So that is when you are trying to target that interacting part. So they are called actually host-dependent or host-directed therapeutic strategies. You can just, right now there are some drugs available, like we can target miRNA and then maybe transcription nuclear factor and metabolism related associated protein interactions, all those can be done. So there are some available targeting host factors and pathway important for viral replication. People are working on these drugs like kinase inhibitor, lipid biosynthesis, and modulating even epigenetic modifications also. Viruses they just modulate methylation and acetylation and all that. So, but these are just at preclinical level, not been approved. So far we don't have any influenza drugs targeting the host protein interactions, right? So all we have is certain neuraminidase drugs and 1 or 2 hemagglutinin drugs and the ion channel protein and the vaccines. They're not very effective against viral infection. So the world has to be ready for the second pandemic, next pandemic, and for that people are working, many people are working to understand the interacting partners. If we can come up with some novel therapeutic and alternative therapeutic strategies where these drugs can target both existing and emerging viral strains also, because the reason I have taken, I already mentioned, these two proteins are highly conserved, 90 to 100% sequence similarity with the rest of the viral strains of different host origin like swine, avian, etc. So that's the reason I have taken these two proteins, and we are trying to understand these mechanisms. Yeah, conclusion is we have to do the proteomic analysis and cross-discipline expertise. That's why we are trying to collaborate with the computational biologists. We are wet lab researchers. We don't know all that, but we need computational biologists who help, for in silico docking, etc. Then all the wet lab research we will be able to do, because all well set established experiment techniques. So we need definitely that interdisciplinary collaborations. Okay, so that's all about this one. Little bit about influenza virus and what my lab is working on, right? And thank you. That's my lab and my students. So that's one, trying to understand host protein-protein interaction, and these protein interactions we can target for developing some drugs. All right, so I have 5 minutes before I left, so now you guys can ask me anything you want.
Or other.
Exactly. So definitely we cannot target the host proteins. You're right, because it plays multiple roles, nucleolin. So directly I cannot target nucleolin. That is why I'm trying to target the interactions. Now I know, if I identify exactly the residue, 3, 4, 5, some residues, they're important, critical for the interaction between the viral nucleoprotein and the host protein. Now, you can make small peptides, tripeptides, tetrapeptides. So these peptides mimic the nucleolin. So we are not touching the nucleolin, we are fooling the virus. Okay, we are fooling the virus, and these peptides will bind to that. Okay, nucleoprotein. So we don't know those because the virus needs some nuclear localization signals, because the virus enters, this nucleoprotein has to enter the nucleus. Okay, so maybe if you just, these are called peptidomimetic drugs. They mimic that particular protein. So you do it, and these peptides will bind to this nucleoprotein. Probably the function will be their translocation from the cytoplasm, from the nucleus, can also be inhibited, or their interaction with, you just make some peptides, nucleolin, because we have seen through RNA binding domain, there is an interaction between the nucleoprotein and nucleolin. So you just block that one peptides mimicking the nucleolin.
But, that peptide which we can synthetically produce in our lab and incorporate into the nucleus, that cannot produce any immune response to our body?
That we all have to take care of all the things before doing. Yes, we have to do it. What immune response we are getting, because that is like a cellular component, logically it should not. Because we are trying to mimic the nucleolin. Nucleolin is the host protein. So we should not expect. Any foreign material will have the immune response. So of course we have to take care of all the things. We need to consider rest of the things. Okay, so that's how we are trying to, because there are peptidomimetic drugs are there. For example, HIV, protease inhibitors. The protease is very important. Okay, so you just peptides, you design peptide. The viral protease thinks it is the original peptide. It will try to just cleave that one, and it gets locked. Okay, they just basically we are trying to fool the virus. Mechanisms. The virus actually does fool the host, but we are also trying to do that one. So that's how we can block the interactions.
The mechanism is likely similar DRD3 mechanism in bacteria.
Okay, okay, yeah, okay.
In that case also DRD3 mechanism, it produces like protein which mimics the nucleotide and जो
exactly, I understand, yeah. block करते हैं, हाँ. Yeah, because they just mimic the host proteins. Because the other reason is the drugs are not successful against viruses. I think by 1940 we got all the antibiotics. Okay, because there is a lot of difference between suppose your ribosomes. Ribosomes are host ribosome and bacterial ribosomes are different. But you cannot, it is very difficult for the virus because virus is using the host machinery. Okay, if you try to develop, as I said, the DNA polymerase block karna hai to, then the host also has got the DNA polymerase because a lot of counterparts will be there. That is why we are not successful with antiviral drugs. In 1960 later, we started randomly screening these chemical libraries. That's how they identified the amantadine and rimantadine drug, and later acyclovir, ganciclovir. I don't know whether you heard of these chemical compounds, acyclovir for herpes simplex virus, ganciclovir for cytomegalovirus, and azidothymidine for HIV. So that's how we came up, because it is very difficult to target the viruses because the virus is an obligate intracellular parasite. It uses the host machinery. Okay, so it's a bit a difficult task, very tricky. So yeah, we need to understand the in detail, So that's why we try to do at the domain level and residue level. Even then, we have to do a lot of things to understand the clear regulatory pathways mediated by these viruses. Okay, yeah. Any other doubts, anything?